Hello, this is my presentation on bioanalytical techniques in therapeutics practicals 4 to 6. The aim of these experiments was to examine actin expression levels in various cell lines, which were NRK and HepG2. This practical occurred over 3 weeks and it was 1 day a week. In the first week, the first stage of the overall process occurred. This was sample preparation of cell extracts, biorad protein assay, and protein standard curve generation. In the second week, the second stage occurred. This was protein separation via SDS page of samples. Also, the third stage occurred in the second week, which was the transfer onto PVDF membranes. In week 3, the fourth stage of this process was the Western blot detection. A fifth stage could be performed if the equipment was present. An example of what could be performed is semi-quantitative analysis of protein bands using computer-based programs such as densitometry. Introduction. Western blotting was first described by Tobin et al. In 1979. Western blotting is the transfer of separated proteins from gel matrix to a membrane followed by subsequent antibody-based detection on the surface of the membrane. Western blotting analysis allows for the detection and identification of a specific target protein out of a complex protein mixture, for example the cell extract. The resulting data allows for both qualitative and semi-quantitative information about the protein of interest. The basic principle of Western blotting is to use 3 elements to accomplish this task. The first is separation by size, the second is transfer to a solid support, and the third is marking target protein using a proper primary and secondary antibody to visualize the protein of interest. Western blotting has been used in the medical industry for numerous diagnosis tests such as HIV infection, hepatitis B virus infection, bovine spongiform encephalopathy, FIV in cats, and Lyme disease. The applications of Western blotting in the industrial setting are to determine the molecular weight of a protein, to measure relative amounts of the protein present in complex mixtures of proteins, qualitative determination of specific protein expression. Some advantages of Western blotting is that it is a highly sensitive technique. As little as 1 to 5 nanograms of an average-sized protein can be detected by Western blotting. I will now answer some of the following questions. Why is it necessary to block? It is necessary to block to increase sensitivity and prevent non-specific signals. What is the function of Kamasi blue stain? The Kamasi blue stain is used as a qualitative test to test for the presence of actin and to determine its molecular weight. Why is Ponkyo red used? It is used as a stain during the reversible staining of transferred proteins and for the qualitative visualization of proteins on the PVDF membrane during immunoblotting. What is the difference between PVDF and nitrocellulose, or when should you use them? Both are used during Western blotting. However, nitrocellulose is ideal for detecting low molecular weight proteins, while PVDF is more suitable for detecting higher molecular weight proteins. Why is it necessary to normalize protein content? The normalization of protein content is carried out to account for differences arising from cell culture conditions during extraction and inconsistent sample preparation amongst everybody in the groups. Therefore, it ensures that the same concentration of a particular cell line's protein is loaded. If normalization is not performed, then inconsistent amounts of protein will be loaded onto the gels. Here is an overflow of the methods used in week 1, which are the protein lysis and extraction methods. First of all, cells were rinsed with PBS. Cell lytic was added and protease inhibitor cocktail. Incubation occurred for 15 minutes on a shaker. The cells were then scraped from the flask. The solution was transferred from the lids to Eppendorf's via a syringe. The flasks were then rinsed with more cell lytic and washings were added to the Eppendorfs. The Eppendorfs were then incubated on ice for 30 minutes. The Eppendorfs were centrifuged for 15 minutes at 4000 xg. The supernatant was then transferred into a chilled second Eppendorf and labelled. The pellet was resuspended in 1 ml of PBS and labelled. A small amount of supernatant was kept for the Bio-Rad protein assay that was performed. Both pellet and supernatant Eppendorfs were frozen and stored for the next week. In this diagram you can see the sample preparation in this step, and the next step involves gel electrophoresis and membrane transfer, which will be described next. Method 2 of week 2. This is the protein separation via SDS page and protein transfer onto PVDF membranes. The supernatant samples were normalized, 2 gels were placed in the tank and the running buffer was added to the inner and outer chambers. The molecular weight ladder, pure actin and the prepared samples were loaded carefully into the designated wells. The gels were ran at 100 volts for 30 minutes. The first gel was kept for PVDF transfer and the second was stained with Kamasi blue for 1 hour. The PVDF membrane was cut to an appropriate size. The gel, filter paper and fiber pads were allowed equilibrate in transfer buffer for 15 minutes. The membrane was placed in methanol for 15 seconds. The membrane was then placed in deionized water for 2 minutes and then equilibrated in transfer buffer for 5 minutes. The gel sandwich was assembled on a cassette and then the cassette was placed in the transfer tank and ran for 50 minutes. The cassette was removed and the membrane was washed with deionized water for 30 seconds. The membrane was then submerged in Poncho red for 1 minute and then rinsed with deionized water until red colour faded away. Next is the Western blotting stage. This occurred on week 3. First of all, blocking was carried out. Then the incubation of primary antibody for 1 hour was performed. The membrane was then washed with buffer and the secondary antibody labelled with biotin was added and incubated for 30 minutes. Next, the extra avidin peroxidase EAP enzyme was added and incubated for 30 minutes. This allows for avidin to bind to the biotin label. AEC substrate was added and incubated for 5 to 10 minutes. The bound peroxidase catalyzes the oxidation of AEC to form a red-brown insoluble precipitate, and this signifies the presence of actin. The membrane is placed in milli-Q water to stop the reaction and then it is left to air dry. This diagram illustrates the Western blotting occurring. Here, a specific antibody binds to the actin of interest. Next, the secondary antibody with a label binds to that primary antibody. Extra avidin peroxidase, or EPA, binds to that secondary labeled antibody, the biotin. Next, the substrate then binds to the enzyme, and that is AEC substrate. And the AEC substrate allows for the detection of actin. Now I will discuss the results. On the left is the Bio-Rad assay, and on the right is the readout result. In the Bio-Rad assay, an initial error occurred in loading. So here are the initial concentrates loaded, and then up here it follows on up until higher concentrations. As the protein concentration increases from low to high, a color change can be seen to occur, which starts at a browny color for low concentration to a blue for high. This correlates well with the data on the right, as it starts out at low at 0.467, for example, and rises up to 0.7. This data was then averaged by everyone in the group and shown in the next table. Here is the table of the Bio-Rad protein assay for each student in Group B. On the left is the concentration from 0 to 12.5, and then the unknown concentration for each individual's protein. This data was then applied to a graph and used to normalize the data. Here is the protein BSA standard curve for Group B. It is noted that point 0, concentration 0 and concentration 12 are left out to generate a more linear graph to give a greater representation of the equation of the line. The equation of the line was then used to determine the unknown concentration of my protein, and that was determined to be 31.345. Here are the two gels while they're still running in the transfer tank. Here, the Kamasi blue stained gel appears. On the right is the ladder. Next is the pure actin band. Here's the supernatant, then is pellet. A space was left. The ladder was then reapplied. Clifford's supernatant, Clifford's pellet, my supernatant, and my pellet. It was noted that overspill may have occurred in this lane. The membrane is shown here, which was taken out and disposed of by error. Due to this error, the membrane results had to be obtained from Adrian, Fidel, and Sam. Here is their Kamasi blue stain. On the left is a pre-stained ladder, the pure actin band, a space, Fidelma's supernatant, Fidelma's pellet, Sam's pellet, Sam's supernatant, Adrian's supernatant, and Adrian's pellet, and then spaces follow. In the PVDF membrane, the red-brown bands can be easily seen, which indicates the presence and separation of actin. The molecular weight of actin was determined to be approximately 37 kilodaltons by using the pre-stained ladder. Discussion. The normalization was conducted of all cell line protein samples. Actin was separated and seen as red bands on the PVDF membrane, and it was determined to be 37 kilodaltons in molecular weight. An optimization to improve these practicals would be to use less BSA standards, as the higher concentration skewed the results of the standard curve, so they had to be removed to obtain a more linear representation, which was required to perform the normalization step. To use dried milk as the blocking agent instead of 0.05% tween 1% BSA and PBS, and also to reduce the quantity of actin required for loading to minimize the overspill effects and occurrence of them. If troubleshooting of a blank membrane with no protein bands or ladder has to be conducted, it may be suggested that poor transfer, incomplete transfer, or overtransfer has occurred. This means that it is vital to ensure that membrane preparation is thoroughly conducted and sufficient gel membrane contact occurs. Also, to optimize the transfer time and reduce the voltage used. For troubleshooting a blank membrane with only a ladder, it may be necessary to increase the concentration of antibody to ensure that the antibody hasn't expired, to increase the amount of protein used, and to confirm the host species. Conclusion. Overall, sufficient identification, detection, and separation of actin was performed. A good understanding of Western blotting process and sample preparation steps was achieved.